introduction, and Jason and Katrina for having me here and inviting me here. So what Katrina already elaborated is, I'm talking about life and death of both virus and host cells. And let's see how I'm doing that. Okay, so one particular aspect which is already quite well known is the influenza life cycle and the required host factors to provide the replication of the virus. And this is just a sketch and schematics from a quite recent review paper by Watanabe. So what you see is you have the cartoon of a host cell. The virus attaches to the host cell, and there are quite some host factors already involved. First steps are the endocytosis to fuse the viral particle with the endosome, which then transported to the nucleus. The viral genome is then released and further transported in the nucleus. Within the nucleus, splicing and replication is commenced by further host genes. The processed and spliced mRNA then get transported out again into the cytosol from the nucleus with other host factors involved. So you have the nuclear export system here, nuclear pore complex. Outside in the nucleus, in the cytosol, you have the ribosome that then translates and produces proteins. They are further transported by the Golgi apparatus, assembled, and then through lipid rafts, the freshly assembled, newly replicated new viral basically buds out of the host cell and forms a new active virus. And what the color code of these host factors that are shown here are so that the yellow are well known. The red one are from also quite recently screens that Watanabe has reviewed in this paper, and I will come to these screens on my next slide. So actually not on my next slide, on my next slide. So the method and the model, what I'm looking at is mostly qualitative. So I'm looking at this Larry Hunter style fastball, which I actually don't want to look at, but I want to use it and analyze it. So this doesn't make any sense really, but my method hopefully does. This just shows even not the whole network what I'm looking at, but just the subnetwork that actually is formed by the second degree neighbors from the viral proteins, and you see some of the usual suspects. You have cyclin-dependent kinase, you have some of the ribosomal proteins, TNF receptor associated factor, so TRAF1, and so forth. And so my method, which I also published quite some time ago, actually makes use of both the experimental data and the experimental data. I will come in 10 minutes. And combines them with genome literature information, mainly, of course, interaction information, so I know which type of components, proteins, DNA, genes, as well as metabolic components and enzymes interact. I need to know the context of these interactions that happen in the cell. Then I assemble everything, come up with my fastball. On the other hand, I have my experimental data, which can be quite from different origin, gene expression data, proteomic data. I could include subcellular locations using protein tags and produce RNA Z-score, which I will use in this talk, to produce pathway scores to identify a so-called functional response network in the context of the experimental data, and the network, which is then technically a subnetwork based on K-shortest path between particular nodes in my take nodes in my network. And this can be, to some extent, of course, used to update and refine the large network. But what even is, of course, more important is to provide feedback to the experiments and actually to make some sense, make a prediction in this qualitative sense, which then further can be experimentally verified. So in a more concise way, using the data from Watanabe as a starting point, and Watanabe used RNAi screens from predicted and from published screens, and he looked and I found 128 influenza host factors that are at least common in 2 of these screens. And again, you see some of the known factors and subnetworks that are required for the viral replication in the life cycle. Here are the viral proteins, and that's why we're looking at host pathogen interactions. Although there are only a few proteins, fewer than the host proteins, here they are interconnected with the human host network. Then some of the first functions that are required for viral replication are endocytosis, fusion of the viral protein, then further the transport of the RNAp, so these are ribonucleate protein complexes into the nucleus. There's splicing within the nucleus. There's then again outside the nucleus in the cytosol translation, but before that happens, the processed RNPs and replicate RNPs are then again transported out of the nucleus. So I have a lot of these functions in this network, but this, as I said, is just one part of the whole system. And what we're really interested in are 2 aspects. The one aspect is we're looking at RNA interference screens. And RNA interference, just briefly mentioned, is a method to screen and to knock out subsequently genes in a host cell. And that has been done in one aspect, looking at the viral replication after infection. And there were particularly 4 papers that have been published in 2009-2010 by Brass, Callas, Koenig, and Shapiro, and they looked mostly at the completely at the host factors for viral replication. And they used different types of host systems. These are lung cells, bronchial epithelial cells, osteosarcoma-derived cells. What's important in my talk is that these 2 are derived from cancer lines. This is a regular cell line which is just immortalized, so which doesn't have anything to do with cancer. And they also use different sets of RNAi targets, between 13,000 and 23,000 almost. And the paper itself looked only at the viral replication in the host factors, but one can, of course, look at the other direction, looking at the proteins, the host proteins that prevent the host factors that prevent the viral replication. So this is one set what we're looking at. And that has been published by others. What we did by our own screen is we're looking at the factors that affect the cell death induced by viral infections. And again, we can look at both sides. We look at the proteins that prevent cell death, so meaning they are sensitize the host cells for infections when they are blocked, and the proteins that are inducing cell death, or the host cells that become more resistant against infections when these factors, these targets are blocked. We are using HBEC cells, so human bronchial epithelial cells infected by WSN, by the one lab strain of H1N1 virus. The others also use the PR8 strain together with the WSN strain. And we use 21,128 RNAi protein targets. So these are the second dimension of our analysis. And when combining both 2 by 2, we have 4 different scenarios, so 4 phenotypes in this sense, and we classified them in these 4 classes. So what you have is we have proviral host factors that facilitate the infection, proviral host factors that deflect pseudo checkpoints, death factors and restriction factors, and just to make that more with some of the main players in this area, to make it more clear. So here would be all the results from this 4 paper with considering any other scenarios. So here the host factors that the virus is required for replication. On the other hand, here would be all the defense factors. So the host cell that are the factors that are required, used by the host cell to defend itself against viral infection. These are the one dimension. The other dimension is these are the genes that would reduce the survival upon each one in one exposure, meaning these are actually the genes that would go into apoptosis, meaning that the host cell would start and initiate this, utilize these genes to initiate cell death. On the other hand, here the classes that would increase HBAC survival upon H1 exposure. These are to some extent very interesting, especially this class, is that they are preventing cell death, to some extent induced by the viral. On the other hand, here used by the host cell against death of the host cell. So we didn't stop there, but actually looked at particular networks in these 4 different classes. And so we used the method to identify the small networks. And again, here the 4 classes. Oh, and I may have to, can I go back? Yeah. So the axis here and so the x-axis is my viral replication, and the nomenclature here would reflect the inhibition due to RNAi screens. So meaning if I inhibit the factors, I would have low viral replication, but these are actually the factors that are required, as I said. Here, high viral replication, high cell viability, and low cell viability after inhibition by RNAi screens. Just to confuse my auditory, I just renamed it here. So these are the viral host factors on the x-axis. On this side of the axis, I have the viral resistance factors, which are the host defense factors. On the other hand, here are all the factors that would work for the survival of the host cell, and here against the survival of the host cell. And again, to reiterate, I have my 4 classes. I have the host cells that would facilitate the infection, actually, and then would kill the host cell. On the other hand, these are the host factors that would deflect the host-induced suicidal checkpoints and would maintain the survival of the life of the host cell. And the interesting thing is, what I'll come into in a minute, is we found quite some interesting pharmacological targets, meaning when preventing these targets, one actually could find some drugs that prevent these targets and then induce viral death. And here are the host defense factors again for death, and now my laser pointer died. And again, other restriction factors. So in detail, for this first class, both in class 1 and 2, one finds the usual functions and usual subnetwork and pathways for viral replication. So there are cell adhesion. These are the V-type ATPase that are required for fusion of the viral cell with the endosome. Furthermore, there are the nuclear pore proteins required of transport into of the viral RNA to transport them into the nucleus. Next, there would be the splicing within the nucleus and the initiation. And as a last step on this network, the transport again from of the newly transcribed RNAs, viral RNAs from the nucleus out in the cytosol. The color coding of these nodes are actually reflecting the particular Z-scores of the RNAi studies, and for the particular quadrant, and the node color refers to the host cell survival axis. The outline color, the border color refers to the viral replication axis, and in the class 1 I would have positive Z-score for the survival and negative Z-score for viral replication. So this would be the common node what you expect. I'm somewhat more tolerant and the majority of these nodes do have this type of nodes, but I'm somewhat tolerant to allow other nodes that don't obey this particular schema in my network, particularly because they may have something to do, also something to do in particular functions, and may actually have multifunctional proteins in this sense. For the class 2, which are more or less all the right color, so both negative in the host cell survival as well as in viral replication. Most of the proteins, so this is the other part what's required for viral replication, which to some extent are the transport of viral vesicles, as well as the huge chunk of ribosomal proteins. And also not quite surprising. And then there's some other subparts, the PARA pathway, for example, which is interesting. On the other side, the host, and these are part of the cell cycles, and actually some of these cell cycle genes, particularly the CHEK1 target, is quite interesting. We found some inhibitor of CHEK1 which actually prevents influenza. I will have another slide for that. So on the host side, this would be the pathway that is important for host defense against viral infection, but this would also induce cell death, so both red, both positive. Proteasome seems to be important. The usual players are all the interleukins. WNT signaling is somewhat new. It's some importance in cancer, but it remains to be seen why this shows up here in this class. So we haven't done any experimental verification studies in this respect yet. And then we also have some other receptors, which also has to be still understood why that is. And again, part of the cell cycle, which functions apparently in both ways in virus replication. And in the fourth class, there's just some basic reformers and not so much what we could identify as importance in this sense. So I come to my last results slide. So as I mentioned before, we looked in particular at this class 2, which apparently have some pharmacologically addressable networks, specifically this CHEK2. And so we looked at the close vicinity of CHEK2 and found, for example, timeless, which recently has been found to be the master regulator of host defense with all respect to viral replication. There's another gene which the CRY1 which regulates of apoptosis, which at least has some importance in plant and plant defense against pathogens. So what we used then is there's a inhibitor, and I think this also was known based on from cancer cells. This is an SB218078 inhibitor that inhibits CHEK1, and by administering this particular chemical compound after 8 hours, the virus actually can no longer be replicated in the host cell. And here they are on this slide, on this figure, you see just the immunostaining of the host cells with and without with for different tissues. And here the importance of the regular cell line compared to the cancer-derived cell line comes into play, because we couldn't find this inhibition in the cancer lines. So this capability is gone in the lung cancer line in the A549 cells, but remains in the HBAC in the regular cell lines. And that's what you see here. The top line looks at the stains the so these are anti-influenza staining, and the top and the bottom just stains is just a regular Höchst nuclear staining, which just stains the host cells, and you see nothing happens in 8 hours without and with administering of the inhibitor in the A549 cells, but in the HBAC cells, there you see some infected cells, and after with a higher concentration, so with the drug, no influenza virus particles can be detected anymore. And in this sense, I will come to my conclusion. So I represented 4 particular phenotypes that characterize important factors for viral replication as well as host survival and defense. And these modifiers can be identified and characterized with the corresponding functional networks and their interaction in these important classes. And we also found that these networks can actually serve as predictors of targets and subnetworks in this important class 2 that actually is used by the viral cell to deflect cell death for continuing replication, but when inhibited with a chemical inhibitor, actually prevents viral infection. This JAK1 is one type or one target what we found that could be used as intervention against influenza infection. And in this sense, I want to acknowledge my collaborators. A lot of people here worked on the experimental side of the RNAi studies. Mike White is the PI of this study, together with Adolfo Garcia-Sastre from Monsignor, who provided some of the influenza samples. We did some other influenza strains too. The people who actually, the students who actually did the work are underlined here. So Sam Ward, who did the experiments on the RNAi. My students, Yuxing Liao, did the combination and statistical analysis of the particular published RNAi screens and combined them with our RNAi screen and did the network work. And Alex Levine, he actually coded a nice Cytoscape plugin to do all the network analysis. And UT Southwestern provides some funds as well as NIEID. And as you may know, the skyline of Dallas in the background, but this guy didn't really contribute, and I want to thank you for your attention. Questions?
I have a couple questions, but that's a very interesting study. So I seem to remember that the 4 published RNAi screen data have very limited overlap. So if that's the case, how did you deal with that when you constructed your network? Second question is, how would you apply this kind of analysis to data acquired in vivo? For example, when people measure blood transcriptomics after infection with influenza, something like that.
Yeah, the first question, that's true. So there's not so much overlap, but as you remember, so Watanabe used at least, if the targets are found at least two of the screens, then we use them as valid. And so that's what Watanabe did. We did some more, so we combined quantitatively all 4 so we refined them, renormalized them, and worked from the RNA scores and combined all 4 individual RNA scores in one score and used that. But Watanabe mostly, so the identified targets were some are listed after. So the targets that are shown in 2 different screens. That's what Watanabe was using. For your second question, yes, RNA. Well, the thing is, you would use so you would work on cell lines and then use the existing or the identified targets on blood samples or other samples, and then of course you have to many other problems to consider if these targets actually show up in the blood. So you have to do some further experiments and check if these are valid targets to be used in blood sample, for example.
Any other questions? All right, let's thank Dr. Forst again.